Now in the last session we discussed how glucose, the universal fuel for every single cell basically, is broken down by various cells, and we basically said the general scheme is that we've got the glucose in the first place, we go into glycolysis, so glycolysis which happens in the cytoplasm of cells, and the product of glycolysis will be pyruvate, and we get two NADH and 2 molecules of ATP. So a moderate amount of ATP, but it's better than nothing. The pyruvate in the link reaction will produce carbon dioxide, and we produce acetyl-CoA. And this acetyl-CoA then of course can go into the TCA cycle in the mitochondria and produce carbon dioxide and a lot of some ATP. And this ATP is produced mainly through the NADH that comes from the oxidation of the acetyl-CoA through the respiratory chain that we have got here. We also discussed that the acetyl-CoA can actually be used to produce ketone bodies. So we might have 2 acetyl-CoA and we dimerize that, or we do a condensation reaction, and in a series of reactions we end up with ketone bodies. And these are produced predominantly in the liver and can also be used as fuels, because this reaction can be reversed and we can use the ketone bodies sort of as soluble acetyl-CoA carriers to various organs. Now if we look at the fuel demands of different organs, we find that for example usually cells, when they've got enough glucose, so we are in the resting state, there's no starvation, so we will find that the brain usually uses lots and lots of glucose. It's the largest glucose utilizer of the body. 60% of all the glucose that we use, that we eat, will go to the brain. The brain doesn't have any glycogen stores. The skeletal muscle, they use very often fatty acids, and they also have glycogen storage. And the heart muscle doesn't use a lot of glucose. Adipose tissue eats glucose, kidney glucose. The liver doesn't really use a lot of glucose. It uses predominantly keto acids because it keeps the glucose for other organs. And very importantly, if we look at erythrocytes, red blood cells, they are solely dependent on glucose. Now if we go into short-term starvation, for in between meals or when we carry out some exercises, we still see that not a lot will change. Erythrocytes need glucose. If we go into longer term starvation, we will see that the brain will switch to these ketone bodies after about 3 days. So after 2 to 3 days, the brain will no longer utilize glucose and instead will rely pretty much on these ketone bodies, which are produced in the liver. The skeletal muscle will also switch to ketone bodies and will rely on the Cure cycle, where we convert pyruvate to lactate, and that lactate is then delivered to the liver, and where it is regenerated into pyruvate and fed back to the cells. Again, we see that the ketone bodies really make up quite a large amount of the fuel for the organs upon short-term starvation. Glucose, however, still remains the only fuel source for erythrocytes, because erythrocytes don't have mitochondria, and erythrocytes can't do anything with acetyl-CoA. And that gets even worse when we are looking at long-term starvation, so a week or so. Basically everybody switches to ketone bodies or ketoacids, but erythrocytes, since they don't have mitochondria, they depend absolutely 100% on the presence of glucose. So where does this glucose actually come from? And of course it comes from the liver. The liver is in a way the glucostat. So liver is the glucostat. That means it keeps the glucose levels in the blood constant. So is the glucostat. And how does it do that? Well, let's go again through the glyco. So we start with glycogen. The liver stores glycogen. And remember, glycogen is the glucose, several glucose molecules, one for, so we've got lots of these 1,4 glycosidic bonds. And we hydrolyze these bonds. Actually, we don't hydrolyze them, we phosphorylize them. So phosphorylize the bonds. So it's basically like hydrolysis, but not with water. We use phosphate for that. So what we get from this phosphorylation, which is catalyzed by the enzyme glycogen phosphorylase, it preserves the energy that is in these 1,4 bonds and converts it to a phosphor bond. So the glycogen will be shortened by one glucose molecule, and I write this as glycogen N-1. So we chop off one glucose molecule and we add the phosphor group through this phosphorylysis, and we get glucose 1-phosphate. So the 1 position becomes phosphorylated, which then, with the help of the enzyme phosphoglucomutase, is converted into glucose 6-phosphate. And I just simply write it like that. Now, in the liver, this glucose 6-phosphate molecule is transported into the endoplasmic reticulum through some glucose 6-phosphate transporter molecules and translocases. And in the ER, the phosphate group is removed through a glucose 6-phosphatase. So glucose 6-phosphatase. It will remove the phosphate, and we end up with glucose. And this glucose then is transported into the bloodstream, where it will be available for the erythrocytes. So that is a system that basically works in the liver, and the liver can really make sure that the glucose level in the blood is more or less constant to feed the erythrocytes. Muscle cells that also store glycogen don't have this transporter that transports glucose 6-phosphate into the endoplasmic reticulum. So muscle cells cannot provide the blood with additional glucose. They are basically pretty selfish. They keep all the glucose from the glycogen that they store for themselves in the form of glucose 6-phosphate. Now, for the erythrocytes, there is an additional level of complexity, because they not only use the glucose as a fuel source, as the only fuel source, they also need this glucose for yet another pathway. This is called the pentose phosphate pathway. Pentose phosphate pathway, or short PPP. Now I should say it's not just the erythrocytes that use glucose for that pathway. It is every single cell has this pathway. It's a very old and, how shall I say, very well conserved pathway that is quite ancient. And this pathway uses glucose 6-phosphate. So we start with glucose 6-phosphate here. But instead of going into glycolysis, as I described up here, instead of going into this glycolysis process with the conversion then to fructose and then pyruvate, instead of doing that, this glucose 6-phosphate becomes oxidized by the enzyme glucose 6-phosphate dehydrogenase. And that is sort of the rate limiting step in this process. And it basically takes 2 electrons away from this glucose 6-phosphate and transfers them onto an NADP molecule. Now, NADP is pretty similar to NADH. So it has the same structure, it just has an additional phosphate group. And why is that of significance? So NADP, if we generate NADPH, cannot enter the respiratory chain. It is not used as an energy source like NADH. Instead, it is used for building other molecules, used for, and here is a list of the things that NADPH is used. The functions of NADPH is widely used in cholesterol synthesis, and you've learned that cholesterol is important for the membrane integrity, for fatty acid synthesis, predominantly in the liver, and also for the reduction of a molecule called glutathione, which protects cells from the oxidizing effects of oxygen. So this ROS here, these are reactive oxygen species, and oxygen itself is a pretty heavy toxin. It's quite toxic to cells because of its oxidative powers. And of course, erythrocytes, the function of which is the transport of oxygen, they need to protect themselves against these reactive oxygen species. So in erythrocytes, we need a lot of this NADPH. So we need to use in erythrocytes, so in red blood cells, lots of glucose is used. Glucose used for protection. So the erythrocytes not only use the glucose as an energy source, but they use it also for protection. And the pentose phosphate pathway generates this NADPH. It also generates 5-ring sugars, riboses, and which then will be used in other steps, for example, for the synthesis, or will be converted for the synthesis of DNA and RNA molecules. So that is the function of the pentose phosphate pathway. In all cells, the erythrocytes are not too bothered about DNA or RNA synthesis because they don't have a nucleus, so they don't need to synthesize anything, but they are really concerned, as much as an erythrocyte can be concerned, about the protection from the oxygen, and therefore we need this NADPH. So it highlights the fact that we really need glucose for the erythrocytes. So where do we get the glucose from? As I said, we get the glucose from our food in the first place, and then the liver will make sure that the blood glucose levels are maintained by using the stored glycogen that the liver stores and that it produces. But after about 16 hours or so, the glycogen is basically used up. And if we didn't have another mechanism that produces glucose, the red blood cells at least would be in serious trouble. So how do we actually maintain the glucose level in the blood over a longer period of time? Well, we actually produce glucose in a process called gluconeogenesis. Gluconeogenesis. So what is gluconeogenesis? Gluconeogenesis is a process whereby we use basically pyruvate and convert it back into glucose. So we are running the glycolysis in reverse. So basically reverse of glycolysis. And in fact, that is pretty much what happens. Where does the gluconeogenesis happen? It happens, of course, in the liver. It happens in the kidney. And it will also happen in some cells of the small intestine. So these cell types are all able to do gluconeogenesis. In the liver, the main substrate actually for gluconeogenesis is not pyruvate, it is the lactate. And we know that this lactate actually comes from the Cori cycle, where muscle and red blood cells deliver lactate that they produced during glycolysis. So here in the liver basically takes the lactate that comes from the various cells and converts it into pyruvate, and from there we go straight into gluconeogenesis. We start gluconeogenesis. A trigger for gluconeogenesis is when the NADH to NAD plus levels, when they are high, so high, this is a trigger. This basically tells the liver we've got lots of NADH here and we need to do something, because basically the overall reaction for gluconeogenesis is we take 2 molecules of pyruvate and we need 4 molecules of ATP plus 2 molecules of GTP plus 2 molecules of NADH. And we convert that into 1 molecule of glucose. This glucose then can be exported and supply the erythrocytes with valuable glucose. And in a way, that should be NADH. So we run the glycolysis in reverse. So remember, we had for the glycolysis, we had glucose is converted into 2-pyruvate plus 2 ATP plus 2 NADH. And what we see here is that we actually need quite a bit of energy to run this gluconeogenesis. So for glycolysis, we get pyruvate and a little bit of ATP and NADH. If we run it in reverse, we have to invest more energy, which makes sense because, you know, we need to get additional energy to run this process of glycolysis in reverse. And we also need NADH. So only if we've got lots of NADH present in the cell, we can afford to run this process. And only if we've got lots of ATP present, we can run this process. Where does the ATP come from? Well, of course, this ATP comes from other processes, like, for example, the beta oxidation of fatty acids, which go into the TCA cycle and which then produces more NADH, which then also goes into the respiratory chain and produces additional ATP. So we are basically in gluconeogenesis running the process in reverse. Well, sort of, because there are some problems with the gluconeogenesis. If we are actually looking at the scheme, it looks a little bit complicated because gluconeogenesis involves 2 organs. It involves the cytosol or cytoplasm, and it also involves the mitochondria. So we need to have both present. And there are actually 3 steps in glycolysis. So if we are looking, here's the scheme of glycolysis. If we are looking at glycolysis, there are basically 3 blocks that where we have one-way streets. So if we just look at the scheme of glycolysis, we start with glucose, and the first block that we encounter here is the conversion of glucose to glucose 6-phosphate. So here, let me indicate that with something red. So here we've got already a sort of a block. The next one-way step is the conversion of fructose 6-phosphate to fructose 1,6-phosphate. That is a block here. And so where it only goes into sort of one-way direction. So here we've got these one-way signs. And the last one is actually the conversion of phosphoenol pyruvate. The phosphoenol pyruvate to pyruvate. And that is a little bit of a tricky reaction because here we would have substrate level phosphorylation, SLP, substrate level phosphorylation, and we need to really reverse this process. So how can we actually reverse this process? And this is actually a process that happens then in the mitochondria. So we basically need to go from pyruvate a little bit smaller, from pyruvate, we need to go to phosphoenol pyruvate. And this is, we can't do it in a straight line. We can't reverse the process in the glycolysis. This is not directly possible. So what we need to do first, we need to do a detour. We add carbon dioxide and we add ATP. And then with the help of a specific enzyme called pyruvate carboxylase, we do something that you probably won't expect. We fix carbon dioxide. Yes, you heard it right. It's not just plants who can fix carbon dioxide. We actually can do that ourselves. We are amazing. So carbon dioxide can be fixed by humans. We also need another vitamin here. We need a compound called biotin, which is also known as vitamin H, which contains sulfur. And from this, with this reaction, we create basically pyruvate plus carbon dioxide. So this would look like CHCO, that is pyruvate. And we add COO to that molecule. So that is our carbon dioxide that we fixed. And this molecule, if that looks familiar, then yes, you are right. This is oxaloacetate. And we have heard about oxaloacetate. Oxaloacetate is the acetyl-CoA acceptor in the TCA cycle. So we generate oxaloacetate in the mitochondria. This reaction happens in the mitochondria. So this is inside the mitochondria. And we produce oxaloacetate. Now, in the mitochondria, the oxaloacetate then is converted to malate because we can never transport oxaloacetate directly out of the mitochondria. We need this malate aspartate shuttle, and we transport the malate into the cytoplasm, where we convert it back into oxaloacetate. And this oxaloacetate in the cytoplasm that we've got now is then used to remove the carbon dioxide again. So we remove the carbon dioxide. We need, however, we need GTP. That's important. GTP, not ATP. And this is carried out by the enzyme PEP, phosphoenol pyruvate carboxykinase. So this enzyme removes this carbon dioxide here. It removes this COO group that the pyruvate carboxylase has attached. In the cytoplasm, this carboxy group is removed, and with the help of GTP, we then generate this phosphoenol pyruvate. So we have to take this detour because the direct step from pyruvate to phosphoenol pyruvate is not possible, at least not in humans. and we convert this pyruvate first to oxaloacetate, transport it out of the mitochondria, and use it there to generate this phosphoenol pyruvate by just decarboxylating the carbon dioxide. So that was basically our first roadblock here that we needed to deal. Now the next roadblock that we are dealing with here, so let's put that here as 1. The next roadblock is this 2, that would be 3. The next roadblock is the conversion of fructose to fructose 1,6-bisphosphate, fructose 1,6-bisphosphate, and we need to convert that into fructose 6-phosphate. And of course, we do that by removing a phosphate group. So we removed a phosphate group, and this is carried out by the enzyme fructose 1,6-bisphosphatase. Now this enzyme removes the phosphate group, and basically it is the reverse reaction of what we see in glycolysis, because in glycolysis we had, with the addition of ATP, we added a phosphate group to fructose 6-phosphate and generated fructose 1,6-bisphosphate. And this was carried out by the enzyme phosphofructokinase 1. So this process here, this is actually, let me indicate that in a slightly different color. So this process here, this is the glycolysis, and is carried out by phosphofructokinase 1, whereas for this process here, for the upper process, this is exactly the reverse process, which is carried out by the phosphatase. So in a way, we've got a sort of a weird cycle here, where we basically shuttle between fructose 1,6-bisphosphate and fructose 6-phosphate. And we use ATP to shuttle in between. So this is where we then look at what we have learned before. We have a system where we have, in one case, we've got a kinase. We have a phosphatase here. I beg your pardon. Let me remove that. So we've got a phosphatase. Removing the phosphate, and here we've got a kinase adding the phosphate. And I told you that this is called, it used to be called a futile cycle, but it's not futile, because what we have here, let me just simply show that as substrate, here we do substrate phosphate. We remove the phosphate. We've got the substrate. And here we have the ATP to ADP, where we phosphorylate it. Here is the phosphatase. Here we've got the kinase. And depending on the activities, we've got a sort of, very often, zero order ultrasensitivity. Where when we plot the activities, all we need is a sort of a minute change in the activity of one of the enzymes. So minute change in activity will result in a switch-like behavior. Switch-like behavior. So if we increase the activity of the phosphatase just a little bit, so phosphatase, just a slight increase will result everything in the fructose 6-phosphate state. So we go just over into the fructose 6-phosphate. However, a slight, only minor change in the phosphate activity, if we go down just a little bit with the phosphatase activity, just a little bit down, will result everything in the fructose 1,6-bisphosphate. So we can make a decision just by minor changes in the activities of either the kinase or the phosphatase, which pathway we go. So this zero-order ultrasensitivity, this what used to be termed a futile cycle, or what I see it as this switch, makes a decision whether we go glycolysis or gluconeogenesis. And in glycolysis, for glycolysis we reduce the activity of the phosphatase. So in glycolysis, the activity of the phosphatase is slightly reduced. So we go here, glycolysis. Whereas in gluconeogenesis, we increase the phosphatase activity, we reduce the kinase activity, and we produce glucose, in this case, which then is used by the erythrocytes. So this is an incredibly clever system, and it allows this process. This process here happens in the same compartment. It happens in the cytoplasm here. Let me indicate that perhaps with this one here. So this here happens in the cytoplasm. At the same time, so we've got both enzymes here, but due to this just minute changes can cause a huge effect, we can make a decision, or the cell can make a decision, whether it goes down the glycolysis pathway or whether it goes down the pathway of gluconeogenesis. And there is, of course, a very fine regulation between these two pathways, because we don't want to use up the glucose that we then make in the cell and send them back into glycolysis. So that leaves us basically with the third state here, with the third block here in the liver. That is the conversion from glucose 6-phosphate to glucose. But we've already discussed that up here, where we said that the glucose 6-phosphate is in the liver, is transported into the endoplasmic reticulum through these glucose 6-phosphate translocases, and where the phosphate is chopped off in the liver by glucose 6-phosphatase, and then the glucose is transported into the bloodstream through some glucose transporters out there. So when we look at gluconeogenesis, we basically can say here in step 1 we had to do a bypass. Oops, that's not the right one. We had to do a bypass of the step. Bypass from pyruvate to phosphoenol pyruvate. In this step here we had the switch behavior, and in the third step we had two different compartments. Namely the ER and the cytoplasm, to basically overcome these roadblocks that lead, stop us from running the glycolysis in reverse. So this is basically the process of the gluconeogenesis that allows the liver. Here's a sort of an overview if you want to have a look at it. That basically allows the liver to run glycolysis in reverse, but we need to be absolutely clear, it will cost energy. So feeding the erythrocytes is a costly process, and we need to make sure that the cells that don't strictly rely on glucose basically don't touch the glucose that the liver makes at a cost. We need to tell the cells, don't eat the glucose. It's a little bit like mama liver is producing the food. So let's say mama liver produces food, glucose for the red blood cells, and for all the other members of the family, it produces the ketone bodies. And of course, we need to make sure that the other cells don't eat the glucose, which is exclusively for the poor red blood cell babies who can only eat the glucose. And how this is actually done, I will demonstrate in the next session. So thank you very much for watching, and I hope you found this interesting.